Presynaptic inhibition is one of the most powerful inhibitory mechanisms in the spinal cord. Pathological changes in presynaptic inhibition are imported in several disease pathomechanisms, for example neuropathic and inflammatory pain, as well as abnormal central pain processing. It is important in spinal cord injury and in central nervous system disorders with motor hyperexcitability. So, when animal models for these disorders are investigated, knowing the strength of the presynaptic inhibition would provide significant information. We present here a method how to assess presynaptic inhibition in a mouse model in Vivo. Presynaptic inhibition is mediated by depolarization of primary afferents, which is induced by GABAergic axonal synapses onto sensory presynaptic fibers. GABA-A receptor activity leads to an increase in the chloride conductance, which then elicits primary afferent depolarization due to the local ion distribution. This depolarization blocks the propagation of action potentials into the axon terminals and reduces their strength, leading to a decreased calcium influx and a reduction of transmitter release. So, the excitatory postsynaptic potentials in monosynaptically excited motor neurons can be inhibited without changing the postsynaptic membrane potential and the excitability of the motor neurons. Volume-conducted potential of this primary afferent depolarization give a direct measure of the presynaptic inhibition in the spinal cord. And these potentials can be measured from spinal cord dorsal roots after stimulation, and these are called dorsal root potentials. Measurement of dorsal root potentials are well established and have